Good afternoon, everyone. I hear myself, so I hope you can all hear me well. On behalf of the Toronto Entomologists Association and the Royal Ontario Museum, welcome to the 10th annual Quimby Hess lecture. It's been a while since we've had this lecture, and I'm really pleased to see all of you here. Kindly put your phones on vibrate, please, if you haven't already done so. My name's Antonia Guidotti, and for those of you that don't know me, I am the president of the Toronto Entomologists Association, and I have the privilege of working here at the ROM. And I'd like to acknowledge now that the site is on what were the ancestral lands of many nations, including the Wendat, the Haudenosaunee Confederacy, and the Nishnawbek Nation, including the Mississaugas of the Credit. We would like to thank and acknowledge all the generations of people who have been stewards of this land for thousands of years. Today, this meeting place is still the house to many Indigenous peoples from across Turtle Island, and we are grateful to have the opportunity to work, play, and live on this land. There are other TEA members that are here today. Chris Rickard, our co-treasurer, is somewhere in here. Oh, there he is, right there. Janet Alexanian is our co-program coordinator. She's not here today, but I believe Kazra Prakash, her son, is here somewhere. I know I saw you. Wave at me. Kazra, where are you? Right at the front here. Excellent. Steve LaForest looks after our field trips, and he's here too. Steve, where are you? Oh, way over there. Okay. Unfortunately, Alan McNaughton, our vice president, got into a car accident on the way here, sadly. He's okay, I think, but he is in hospital because the airbag's deployed. So sorry for Alan. He was really looking forward to this. And Calder Forbes, our co-treasurer, is sick today, so she could not make it. Albert, where are you, Albert? Thompson? Yes, our secretary is here as well. This talk is going to be videotaped and recorded so that it will be available online, I believe, on the ROM website and also on the TE one after this. I'd like to say a few words about Quimby Hess, if I may. This lectureship was established in his name and is sponsored by his son, Robert Hess, who is here. Robert is right there. Wave, Robert. And Quimby's daughter, Jane, who is also here. Where are you, Jane? Oh, over there. And Jane is here. Thank you for sponsoring this lecture. It's always been my pleasure to help to organize it and invite a speaker and get people here. It's wonderful. When I first started at the ROM, one of my first jobs was databasing butterflies. And to be honest, I still do a lot of databasing, but some of those ones were donated by Quimby Hess, and I was very fortunate to meet him on one of his visits to the collection. What I did not know at the time was that he was one of the founding members of the Toronto Entomologists Association. His name appeared on the first membership list from March 1969. He remained an active member until his passing in 2010, contributing to the association for more than 40 years. He was a true leader in our community. He served as president for a few years, but he was also involved in a lot of other activities. He played an integral role in many of its major initiatives. One of his most significant contributions came in 1975 with his groundbreaking publication on the West Virginia white butterfly. At the time, this rare species, they didn't know very much about the biology, they didn't know much about the distribution. And his work demonstrated how forest management practices could be altered to allow it to prosper. And this was a pivotal moment in the club's commitment to insect conservation. Quimby would be pleased to know that the West Virginia white is now doing well, just as he had hoped. In 1991, Quimby also spearheaded the book, The Ontario Butterfly Atlas, with a few other members of the association. And at a time when technology was really limited, what they did is they meticulously mapped the distribution of every butterfly record in Ontario by manually pasting dots on a map. Just imagine that way back in the early 90s. These days we can do that really quickly, right, with a lot of different programs. It was the first time people could see the distribution of butterflies across the province, and it was one of the first publications of the TEA. Today, Quimby would be astonished to see how his efforts have evolved into interactive, computer-generated maps on the TEA website. And it's now based on more than 500,000 butterfly records. And if you haven't seen our website, it's ontarioinsects.org, and it's worth having a look at these maps. The Butterfly Atlas was a project that perfectly suited Quimby's dedication to butterfly observation. Throughout his distinguished career as a geologist with the Ontario Department of Lands and Forests, Quimby recorded butterflies from virtually every corner of the province, from Point Pelee in the south to the Hudson Bay in the north, to Manitoba in the west, and to Quebec in the east. While many TAA members focus on the local area where they live, Quimby could have proudly echoed the words of singer Stompin' Tom Connors and a few others who have written, I've been everywhere, man. I've been everywhere. This lecture series serves as a fitting tribute to Quimby Hess and to the remarkable legacy of his lifetime passion of insects and involvement with the TEA. I'd like to thank Jane and Robert for their generosity in supporting this lecture. We are pleased that many of the Hess family are able to attend today, and we look forward to hearing from our speaker. Right now, the curator of entomology here at the ROM, Doug Curry, is going to come up and introduce our speaker, Steve Marshall. Thank you.
Thanks, Antonia, and welcome everyone. What a great crowd coming out on a not so sunny afternoon. So Steve Marshall. Steve Marshall is a curator emeritus at the University of Guelph, where he continues to study the taxonomy of flies, even though he's been retired since 2019, after 37 years of service. Sort of as a fellow dipterist, that's sort of a person who studies flies, I've had the opportunity to follow Steve's career for, more than 40 years. I'm starting to get old. And so, whereas I study black flies, Steve's area of specialty are spherosphaerids, known as the small dung flies. And I guess I kind of consider us kindred spirits from the respect that we both study organisms that have unsavory feeding habits. Steve has really developed one of the great insect collections in Canada at the University of Guelph, numbering at over 3 million specimens at last count. Maybe it's more now. There he continues to teach, or had taught courses in insect diversity and biology. And he's also been a major trainer of students. And so he literally, across North America, there are many specialists, mainly fly people, but also hymenopterists at major institutions across North America. So that's a really good sign of a professor. And all the while, he managed to churn out over 200 scientific publications, which is really incredible. Probably for most of the folks gathered here, you're probably most familiar with him through the publication of some of his biodiversity books on insects, the most recent of which, on Hymenoptera, won the Dartmouth Award for Best Reference in 2024. And here it is here, available at quality bookstores. So please check it out. Of course, he had, my personal favorite is the Diptera one, but there's also one on Coleoptera. So Steve has been a recipient of many awards. For example, he won the gold medal from the Entomological Society of Canada. He won the Thomas Say Award with the Entomological Society of America. And for the Dipterological Society, he won the C.P. Alexandra Award. All prestigious. But to me, the most amazing fact is that he has described over 1,000 species of insect, mostly flies, including a lesser dungfly named after a former prime minister who will be nameless. Please join me in welcoming Steve Marshall to the stage. Steve.
Well, thank you very much, Doug, and thanks to the Hess family for sponsoring this series of lectures. It's a great honor to be invited to give the annual Quimby Hess Lecture. I understand that Quimby's passion for natural history and entomology was ignited when one of his public school teachers brought a Cecropia cocoon into his Southern Ontario classroom. And when one of these beautiful moths emerged from the cocoon, I guess he was hooked. Well, I had a similar experience as a child, but the Cecropia cocoons that I brought into my classroom didn't yield any nice moths at all. Instead, they yielded these really interesting flies. They probably were one of these 3 genera of Tachinidae, parasitic flies, and almost certainly the upper left one here, it's Compsognathura concinata, which was absolutely hammering Cecropia populations at that time. It was deliberately introduced for biocontrol of sawflies, but things went a little off the rails. Of course, as a child, I had no idea of that. I'd never heard of a tachinid, and this was my first exposure to parasitoidism. But what I knew was I'd reared these funky flies, these exciting, unexpected flies out of a cocoon, and it sticks in my memory as a discovery. It felt like a great discovery. Well, of course it wasn't. The parasitoid complex of giant silkworm moths was well known then and is well known. However, the life histories of the great majority of insect species are entirely unknown. Furthermore, the life histories of a great number of genera, or perhaps most insect genera, are unknown. And there's even a good number of insect families for which we don't know the life histories. Here, for example, are 8 families of flies in the group that I study, Acalyptrate Diptera. All 8 families are essentially unknown when it comes to natural history. The larvae are unknown for all 8 entire families. And 2 of these families, including the Inbiomyidae shown here, were only described as new to science in the last relatively few years. This one was described in my lab in 2006. Every species was new, 11 new species, a new family. Well, there's not very many brand new, entirely new families of insects out there awaiting to discovery. Maybe there's a few, but not many. However, there's an enormous number of genera. There's lots and lots of genera that haven't been named or discovered yet. And by way of illustration, these pretty flies are all undescribed species in an undescribed genus of stilt-legged fly that I'm currently working on. So you can see there's a great deal of exploration to do in the world of insect diversity. And what I'd like to do today is do a little bit of that exploration with you, starting off on some well-trodden paths and by looking at what we know about insect diversity, and then venturing a little further into terra incognita by mentioning a few things that we don't know about insect diversity, or at least what I don't know. And then we'll spend the bulk of our time actually looking at some of the major players in insect diversity, asking just what do they look like and what do they do? So first of all, what do we know? Well, thanks to a series of recent authoritative multi-authored publications put out on behalf of the Biological Survey of Canada, we know that Canada has almost 10,000 named fly species, and flies are the order Diptera, almost 9,000 named wasp and related Hymenoptera species, and a few more than 8,000 species of known species of beetles. another 12,000 in the other orders. But today I'm going to ignore the other orders, the true bugs and the moths, and instead I'm going to focus on these 3 mega-diverse orders. These, though, are just the known species, and we don't know all the species in Canada. And those same papers, oh dear, I've got an issue here. We have a problem with a technical problem. We're not advancing, excuse me. What does the message say? Software update. No! Go away, cancel. Okay, let's see what happens now. Oh, that's much better. Okay. Yes, as I was saying, we don't know all the insects in Canada, and those same authoritative papers suggested that when we take into account undescribed or unrecorded species, in actual fact, we have somewhere between, roughly, between 60 and 70,000 insect species in Canada. And they break down, according to those authors, more or less like this. So the beetles come up a little bit from 8,300 to an estimated 9,000. The wasps come up a lot to 19,000. And the estimate for flies was 15,000, but those papers didn't take into account some really poorly known groups of tiny midges, which will add thousands more species. So this sort of summary of the numbers is what you see here. So about twice as many flies as beetles, twice as many wasps as beetles, and these 3 big orders together making up 80% of all insects. And by the way, the examples that I've used here, in case you're wondering what they are a parasitic fly, a tachinid, a trichopoda, which is a parasitoid of stink bugs and other true bugs, an interesting member of the large parasitic wasp family Ichneumonidae it's a hyperparasite. It parasitizes other parasites. And then a beautiful big sexton beetle or burying beetle, and this is Necrophorus americanus. And it was kind of an ironic choice to illustrate insects of Canada because it doesn't occur here anymore. It's extirpated. It was here, and our last specimen at University of Guelph is from the 1970s, collected by my friend Rob Ruffley, but it's gone, so there's an ironic choice there. Well, that proportion between wasps and flies and beetles carries through for the insect faunas of other well-known countries, and the best-known country for diversity in general and insects is, of course, Great Britain. And here we see smaller numbers, somewhat less than half of the Canadian numbers, but almost the same proportion between flies and beetles and wasps and beetles. And again, about 80% of the total fauna, which I think is about 24,000 for Britain. And the examples I've used here are probably things most of you recognize. Some of you are probably going, why is he using pictures of Ontario insects to illustrate Great Britain? Well, it's because these are all things that occur both in Great Britain and in Ontario, and 2 of them, the 7-spotted lady beetle and the European paper wasp, are relatively recent introductions from Europe. The third one, Dryomyza analis, no common name, is probably naturally holarctic. So the picture for Great Britain and the picture for Canada are quite similar. Now, what happens when we look on a world scale? What happens? How many insects are there globally? That's actually quite a loaded question. There's a simple answer that's wrong, which is, here's what we know about insects globally. These are the known, the named species in the world. And if you're astute, you've probably noticed right away that things are reversed. We've got twice as many beetles as we have, either more than twice as many beetles as we have flies or wasps. The beetles add up to more than the flies and wasps put together. That's amazing. Are things that different in other countries? I don't think so. Those of you who have been in my classes, I recognize a few of my ex-students here are sort of going, yeah, martial on again about beetles. What I always say in my classes is that number refers more to the number of coleopterists than the number of coleoptera. And this is really, it reflects the fact that beetles are relatively well known. A lot of people have studied them. After all, it's more attractive to collect and study a shiny beetle than some tiny little soft-bodied fly. And the other way to put that is that just flies and wasps are grossly understudied. And if I can translate that to an estimate of what the world fauna really looks like, and don't worry about the precise numbers here, what I'm trying to get across is an idea of ratios. I think this is more or less what the world of insects really looks like. 1 million or more flies, 1 million or more wasps, and probably, when all is said and done, about half as many beetles. I think that the ratio globally isn't going to be that different from what it is in Canada or Britain. Well, I can't get into detailed analyses of the diversity of beetles and the diversity of wasps today. I have to concentrate on just one selected order because we only have so much time. And I'm going to home in on my favorite order, the Diptera, the true flies, like this snail-killing fly from Central America. It's the order Diptera, and I suspect there'll be a few people here who don't know what that order is or what defines it. Not many, but there might be some people who need to be reminded that the Diptera are the two-winged insects. So they differ from other winged insects. Oh, and 99% of all insects belong to winged orders. So they differ from other insects in having only a single pair of wings. The hind wings are reduced to little knobs called halteres. Other insects have 2 pairs of wings. So the Diptera are extremely important for a number of reasons. They're underappreciated pollinators, but critical pollinators. They're extremely diverse parasitoids, like the South American tachinid fly. There's a lot of predators in the Diptera, like this Cynosia muscid feeding on a fruit fly, or this Lafria robber fly feeding on a deer fly, or these little Dolichopotidae feeding on springtails. The order is of tremendous importance in decomposition. Super important as decomposers, like those blowflies. Less important as plant pests, but there are still some major plant pests in the group. And of course, as we are all painfully aware, this order includes a lot of familiar blood-feeding flies like the stable fly, deer fly, horse fly, black fly, and of course, mosquitoes, which are all of which are of great importance, not only as nuisances, but as vectors of some of the most important diseases facing mankind today. So flies are really important, and that's sort of a simple take-home message. I want to go on from that to talk about diversity, because that's really the topic of my lecture today. So they're important. How diverse are they? I'm suggesting that the global total is around 1 million, but I've described it here as a guess, because to be honest with you, all of these big numbers for world totals for the various orders are little better than guesses. They're educated guesses. This is based on the fact that a large number of insect groups that I've looked at very closely have had between 80 and 90% of the species turn out to be new. So a novelty rate of 80 to 90%. That combined with expert estimates of the total diversity in their particular families lead to a reasonable estimate of about 1 million fly species worldwide. But it's a guesstimate, or rough estimate. The numbers of other insect species globally are also just guesses. Entomologists have converged in the last few years on numbers of about 5 to 6 million, but published estimates in the last decade have ranged from around 2 million up to tens of millions, including millions of cryptic species recognizable only by sequencing. But the general consensus in recent literature is sort of 5 to 6 million. I actually favor an estimate of about 3.5 million. I'm on the low side because I favor the idea that Canada's share of global insect biodiversity is pretty close to 2%. And if our counts of Canadian insects are about right at 60, 70,000, that translates to a global fauna of about 3.5 million. And these particular examples here are pretty close to that 2%. Mosquitoes, we have exactly 2% of the global fauna. Butterflies, I think it's 1.7. Ground beetles, it's 2.5. So these are all rough estimates. The bottom line is it doesn't matter whether there's 3 million, 7 million, or more, that the mission that faces taxonomists like me today is to sort out millions of insect species, mostly flies and wasps, and mostly in the tropics. And we've got to get names on them and get them properly classified, because as Linnaeus said, if you don't have names for things, your knowledge of them will perish. So this is an important mission. And since most of the action is the tropics, let me jump now to a tropical survey that I had someone involved with. This survey was actually organized by Canadian dipterists Brian Brown and Art Borkant, who arranged for hundreds of thousands of specimens to be collected from a private farm, a 4-hectare farm just outside San Jose, Costa Rica. And in that little 4-hectare plot, they got hundreds of thousands of specimens, and they sent some 50,000 of them out to a network of top collaborating taxonomists. And all of these specialists identified the species in their particular area of expertise and came up with, so far, about 4,500 species. So 4,500 species of flies just from a 4-hectare plot. That's staggering. That's, as I say here, it's almost half as many as there are species as there are flies in the world. So there's a lot to explore in dipteran diversity and fly diversity. Now, if we drill down and look at the particular taxa, the families that made up that diversity, of course, some of the big players were the families that most of you know well as attractive, large-bodied, diverse flies, like the Tachinidae, a huge family of 10,000 species worldwide. We got 286 species of tachinids. Flower flies, everybody knows and loves the Syrphidae, about 93 species. But the contributions to diversity made by those big familiar taxa was absolutely swamped out, dwarfed by the input from a number of large families of small-bodied, very poorly known, taxonomically neglected flies. And the best example of that is this family, the Cicetomyiidae. One of our collaborators, Matthias Jassoff of Sweden, studied the cecids, the gall midges, and found 800 morphologically distinct species. So he was sorting them out on male genitalia. That's what we morphological taxonomists normally do. Details. He sorted out 800. There was 10 species known from Costa Rica before. Unexplored diversity. And we look at more of these big, poorly known families. We see impressive numbers over and over. The Phoridae, the scuttleflies, 400 species at Zerkee. Fungus gnats, 268 species. Dark-winged fungus gnats, another couple hundred species. Biting midges, a couple hundred more. On and on. What we see is a general phenomenon of taxonomically neglected families of small flies contributing disproportionately to biodiversity and virtually all unknown biodiversity, because most of the species that make up these numbers are unnamed species. They were sorted out as morphospecies. So I find this to be an important phenomenon and something which is fundamental to the issue of exploring insect diversity. And this convergence of diversity dominance and taxonomic neglect was nicely captured in the title of a recent paper. And the authors of this paper used high-throughput sequencing to sort out about 250,000 specimens from malaise traps all around the world. And the remarkable thing that they found was, even though there was some 500 families of insects in the trap samples they sorted, just 20 species accounted for I'm getting tongue-twisted. Just 20 insect families accounted for more than half of the local species diversity. So I want to talk about these 20 families because they're biodiversity superstars. But before I go in and talk a bit about those families, I'm going to step back and comment on malaise traps, because this survey was done using malaise traps. Some of you are probably familiar with them. If not, let me point out that a malaise trap is kind of like a pup tent with no sides, but with a center panel. Insects fly in the sides, they hit that center panel, and they're directed up to a trap head at the top. Or in some cases they drop, in which case you can collect them in pan traps underneath, which is what I do actually. But the people using these traps as biodiversity sampling devices generally just use the trap heads. And that was what was done in this study. So it's a bit biased. It's not sampling all insects, but the material studied here, the material assessed, is a pretty good representation of insect diversity. Here are the 20 families that those authors found. So I just gathered up my pictures of these 20 families and put them together into what, to me, is biodiversity. To paraphrase E.O. Wilson, these are the little things that run the world. So more than half of local biodiversity, as measured by malaise traps, is represented by these 20 families. And I'm going to simplify this picture a bit and eliminate the families that aren't little flies and wasps and give you a simple take-home message. Biodiversity looks like this: mostly little flies and little wasps. I'm going to talk only about the flies, as I pointed out before. I was going to stick mostly with that one megadiverse order. But before I erase the wasps, let me point out that family number 4, and the numbers refer to the relative contribution to biodiversity. So the first row contributes more to the numbers assessed than the bottom few rows. But number 4, very diverse group, tiny wasps in the family Platygastridae. And depending upon how you define that family, it's either all or mostly parasitic on family number 1. So with that, let's get rid of the wasps and zoom in on the flies. You know, I was going to talk about all 10 families, but I think that's a bit much. So let's trim things a little bit more and just look at the top row, the biodiversity superstars, and talk about them, and also family number 15, which happens to be a family that I work on. So let's dive into that. Number 1, sometimes contributing as much as 20% in various ongoing biodiversity studies, is the so-called gall midge family or cestid family, Cecidomyiidae. And these are really, really taxonomically neglected flies. There was a relatively recent catalog, 2021, by Gagné and Jashoff, and I might interject that Raymond Gagné turns 90 next year, and Jashoff no longer works in entomology. Those esteemed authors, definitely the top experts in the family, introduced their catalog with this statement: the number of species in this family is inestimable. That's quite a number. Despite their admonishment, I'm going to go on and sort of estimate the fauna, because we need some sort of a number. They cataloged about 6,500 species. And Jashoff and Jashoff looked very closely at the cestids of Sweden. Actually, they worked for decades on the Cecidomyiidae of Sweden. And they published an estimate of 1,800 for the cestids of Sweden, along with a statement that this is the only reasonable estimate for any country in the world for the family Cecidomyiidae. So I'll take them at their word. 1,800 species from Sweden. You can sort of extrapolate that up to about 2,500 for Canada, because our fauna is about 1.4 times the Swedish fauna. And you can extrapolate from that up to an estimated world fauna of 125,000. 125,000, that's an enormous number for one family. Remember that the whole fly fauna, as currently known, is 160,000, so enormous. And enormous though it is, other people have come up with bigger estimates. Hébert et al. In 2016, based on barcodes of Canadian flies, estimated that there's 2 million species of cestids in the world. 2 million! That's enormous. Well, I'm not going to debate where in that spectrum between 125,000 and 2 million the real number lies. I'll simply summarize that by saying, we know virtually nothing about this hugely important family. At minimum, 95% of the world species are undescribed. Now, despite that, you probably recognize some. You know, some groups are well known. Some of the groups that make conspicuous galls have been recently revised, and they're well known. So the gall makers induce these particular structures on their host plants. And here are 4 species of galls, cested galls, that I photographed in my backyard this last August. 1 on pussy willow, 2 on tall goldenrod, and 1 on Rudbeckia. And here's what the adult fly that induces the gall looks like. This Asphodila Rudbeckia conspicua that induces big golf ball-sized, multi-chambered galls on Rudbeckia. But it's a relatively large fly. Most gall flies are minute, so you can sort of see now why the family's not that well known. They're pretty inconspicuous. This, by the way, is a serious pest. The Swede midge, one of 2 pest Contarinea species that are serious pests of canola in Canada. This one was just recently introduced and identified thanks to Raymond Gagné, who I previously mentioned. Well, the gall midge family includes a lot of plant-feeding species, mostly unidentifiable, like these females laying eggs on, this is a grass seed head in my backyard. So it's very, very tiny flies. There are some well-known predators in the family. You can even buy Aphidoletes as biological control agents to control aphids. So the orange larvae here are Aphidoletes larvae feeding on aphids on cup plant in my backyard. There's an adult laying eggs on the same plant. But most cestids, or at least most of the undescribed cestids, including 5 of 6 subfamilies in the family, are fungus feeders, and you're most likely to find them laying eggs in rotten wood like this one is. And these are the virtually unknown cestids. It's a truly unexplored part of the planet's diversity, and there's hardly anybody working on them. If you want to look for adult cestids, by the way, a good place to look is sheltered spider webbing. I've found everywhere in the world that I've looked, every country I've ever collected insects basically, I find cestids hanging from spider webbing. And I've asked my colleagues why they do it, my cestid expert colleagues. They say they don't know, but there's lots of species that do this. I also sometimes find them feeding on food being consumed by large predators like this spider in Borneo or this phalangid in Costa Rica. So some of them are kleptoparasites, food thieves, which is kind of cool. All right, so that's the biggest biodiversity superstar according to that particular paper I'm working from. Number 2 is another group of really, really tiny flies with about the same number of described species, same number as named species as in the gall midges. This is the Ceratopagonidae, or biting midge family. The world expert on Ceratopagonidae recently told me that he estimates the world fauna to be, that was Art Borchardt, a Canadian Art Borchardt. He estimates the world fauna to be over 70,000, which says that 10% or less of the species in this family are described. So it's very, very poorly known group. Despite the fact that 90% of the species are undiscovered, undescribed, those of you who like to poke around fresh waters have probably seen these common predaceous species, a couple of common predators in the group. And most of you, if not all of you, have encountered the biting midges in the genus Culicoides. Unlike other Ceratopagonidae, this genus is pretty well known. Big genus, 1,400 species. It's important because for medical and veterinary reasons, some major livestock diseases are transmitted by Culicoides. But from your perspective, these are the almost invisible flies that pass through your cottage window screens and make you feel like someone's taking a hot match and sizzling your arm with it. And local names for these include punkies, no-see-ums, and there's more, but I can't say them in a mixed audience, so we'll go on. Those aren't typical. Those aren't typical of the Ceratopagonidae. The majority, including most of those undescribed species, don't bite people. They bite other invertebrates, either as predators in some cases or as parasites, like this Trichopogon feeding on the blood or the hemolymph of a crane fly, or these three different subgenera of Forsythia feeding on green lacewing in Ontario, or a damselfly wing, again in Ontario, or on a stick insect, and that one's in Costa Rica. And this is such a common phenomenon that if you go home and look at your macro photographs, and I'll bet you many of you take photographs of insects, I'll wager that some of your photos have been photobombed by ceratophragonids, like this pygmy blue and these other insects here. So pretty common. And if you want to go out and actually see a ceratophragoniid in action, the easiest place to do that is on a blister beetle. So next time you see a blister beetle, these are beetles that are loaded with a blistering agent called cantharidin. Their blood is full of cantharidin. Next time you see one, watch it for a while, and I'll bet you'll see these little flies coming in and feeding on that cantharidin-laced blood. And these pictures are from Ontario and South Africa, so this is a widespread phenomenon. Other Trichopogon are common pollinators, and some are kleptoparasites. Now, I have a special interest in kleptoparasitism. Kleptoparasitism is food thievery, and for some reason, I just find it fascinating. So let me take a wee digression and tell you about some kleptoparasitic ceratophragonids that I found. And this discovery took place when I was in Ecuador some years ago teaching an undergrad field course in entomology. And I took a group of undergrads, incipient entomologists, to this termite carton nest. This is a common kind of termite in the neotropics that I've used for demonstrations with students many times. If you cut them open and expose termites, you usually get a common assassin bug, a reduviad, that comes in and feeds on the exposed termites. Now, I wanted to show my undergrad students this phenomenon, but when I cut the termite carton open, instead of an assassin bug, I got these comb-footed spiders. They came swinging in, Tarzan style, on a silken thread. They grab a termite, swing back out, wrap the termite up in thread, swing back in, grab another one, wrap it up in silk. And as this was going on, now that was pretty cool in itself, I got excited, but as it was going on, I saw these little flies come zooming in and feeding on the termites. Then I got really excited and started jumping up and down. I went, wow, kleptoparasitic ceratophragonids, this is so cool. And I suggested to the students that this is a new system. Take some observations, make it your undergrad project, and we'll see what we can do with that data. We brought the data home and the specimens, shared them with a ceratophragoniid expert and a spider expert, and together we published this paper on 4 species of kleptoparasitic ceratophragonidae and this new termite spider system. I thought that was fun. But if you want to see kleptoparasitic ceratophragonidae, you don't need to go all the way to the Ecuadorian Amazon. I took this photograph just this past September, in Algonquin Park. And this is the same genus we found in Ecuador, but this is feeding on a midge, a chironomid, in a spider web in Algonquin Park. And that's convenient because Chironomidae is number 3 on the list of biodiversity superstars that I want to illustrate for you. So chironomids, midges, this is a group familiar to, I think, every Canadian. They're very, very common, non-biting flies. And it's actually the largest family of flies in Canada by known species. So we have about 800 known species in Canada. But chironomid experts, and I'm definitely not one, but my colleagues estimate the totals in Canada to be more than double that 800. And I find that really amazing because chironomids are everywhere. Here are some chironomids over Lake Ontario, and my son Alex there took this picture of some old guy out fishing in the middle of the lake. But chironomids are extraordinarily abundant, and the hemoglobin-loaded larvae of chironomids, called bloodworms, can often be extraordinarily abundant in the mud along Lake Ontario. So they're common insects, familiar insects, and important. If you go in freshwater systems, quite often the dominant group in terms of numbers of individuals and numbers of species are Chironomidae, aquatic chironomid larvae. It astounds me, given this importance and abundance, that more than half of the midge species in Canada are unknown to us. They're not recorded or not named. It's astounding. So midges are a bit hard to identify for that reason, but some are recognizable by distinctive habits. And just to show you a couple of pictures taken recently, this is a species where the larvae mine in floating leaves like water lilies and water shield. And here's one which you can always find in pitcher plants. If you take an eyedropper, take the water out of a pitcher plant, Metrius nemus nabi is always there. But most chironomids, just a chironomid you pick up with a net or you get out of a trap, you're not going to be able to identify it, not unless you're a specialist. So those first three families in that biodiversity top 20 capture the essence of what I want to say about dipteran diversity and the unexplored nature of dipteran diversity, because in these three families we're looking at tens or hundreds of thousands of species and less than 10% known to science. Some entomologists have come to refer to this kind of taxon as dark taxa, because they're kind of biodiversity's dark matter. And you'll often see that term, dark taxa, used to refer to this kind of family. So let's go on and just deal with a couple more families. Even though I've dealt with the biggies, let's deal with a couple more because they are interesting. The Psychodidae, or moth flies. You probably know this family as the group of tiny moth-like flies that you will find flying around your bathroom from time to time. And honest, not a word of a lie, on the way to give this lecture, we stopped for breakfast on the way here. I won't tell you where. And I went in the washroom, and there was Psychoda alternata on the wall. So here we go, topical observation. That's the little one. The big one, Clogmia albipunctata, is more common in institutional washrooms. You can look around today after you're done. But most psychodids are not like the common domestic moth flies. Most of them are really diverse. For example, to illustrate that point, the top two images here are Mariuina, an aquatic psychotid genus, and these are insects of fast-flowing cold water, and you can see in the upper right that the larva is kind of it's flattened like a sucker disc to stick on rocks in waterfalls and fast water. And these bottom two flies are a totally different subfamily, the subfamily Phlebotominae, and these are the notorious vectors of leishmaniasis. That gorgeous fly on the lower right is a phlebotomys from Madagascar, and phlebotomys is the old world vector of leishmaniasis. Lower left, that blood-fed female on somebody's arm in Costa Rica is the vector of leishmaniasis in the New World. And I've got to say that's the fly I dislike the most. I mean, I like all flies, but I dislike that one the most because cutaneous leishmaniasis, kind of flesh-eating disease, not nice, is considered to be kind of an occupational hazard of neotropical fieldwork right now. And a number of my colleagues, I've had the misfortune of getting that. Well, here's another biting psychotid, but this one won't give you leishmaniasis because it only bites frogs. It might give frogs something, I don't know. Let me go on to just one more moth fly, one more psychotid. This is my favorite psychotid, the slime ball psychotid. It's my favorite because it's a recent discovery. And a few years ago, I was walking through a national park in Costa Rica with my son, Stephen. Stephen pointed up into the canopy and said, Dad, what are those glistening blobs up there? And I went, I don't know, let's pull them down, have a look. And they turned out to be root tips with these glistening gelatinous masses covering the tips. And I looked at them and went, holy cow, there's fly larvae in here. I've never heard of anything like that. And fortunately, I had a collecting permit because I was in Costa Rica for the Zerkee survey at the time. And I was able to clip these and bring them back for rearing. And I got the material into the hands of my good friends and colleagues who happen to be the world's experts on Psychodidae. And these flies turned out not only to be a new species, but a brand new genus. So the genus Mucomaia, and a first discovery of this kind of aquatic insect in the forest canopy. So the nice thing about exploring insect diversity is every time you turn around, you find something cool. All right, so one more in the top 20, and I had to slip this one in because this is the family that I do research on, and it's the family Sphaeroceridae that Doug unkindly referred to as small dung flies. They're actually much more than that. These inconspicuous but ubiquitous little flies do develop as microbial grazers. So the larvae get into a lot of things where there's bacteria. That includes dung, carrion, and many, many other things. But the family is actually really diverse. These, for example, are kleptoparasites, the little flies on the back of this elephant dung-rolling beetle will ride on the back of the beetle until the beetle buries its precious dung ball, and then they scoot in and lay eggs in the dung ball, and the larvae develop as food thieves, well, poo thieves, kleptoparasites in the nest. And there's actually 3 genera that do that, one just recently described. And there are other genera that are only found in seaweed. This genus, it's a worldwide genus. It can be found anywhere in the world where you get those big piles of rotting seaweed called wrack, typically along rocky shores. And millions of the larvae of this genus sort of facilitate the rapid decomposition of the wrack and its transformation into soluble materials that go back into the marine ecosystem. And here's an esoteric genus that we recently discovered in bamboo stipules. It's the only place it occurs is in bamboo stipules. The nice thing there's a number of nice things about Sphaeroceridae that have led me to work on them. One is, despite their nondescript appearance, they're like little black flies, they're super rich in complex internal characters, the kind of features that taxonomists like to use to do phylogeny and work out zoogeography and do taxonomy. But also, they're easily collected in huge numbers using simple traps like pan traps. The little black dots in this pan trap, ignore the big shiny beetles. The little black dots in there are probably all sphaerocerids. And using this kind of technique, applied not only by me, but also by many colleagues, we've been able to assemble a comprehensive collection of about 1 million mounted sphaerocerid specimens at University of Guelph. And that has allowed me and my students to go through and revise every genus of sphaerocerid that occurs in Canada, and most of the genera are found in the New World. So you can see how our knowledge of this group has changed over the years. When I started in '79, there was 35 species. Now we have 2,017. On a global basis, well, we still have a lot of work to do. There's still, we know almost 2,000, probably another 8,000 remain to be named. And I mentioned that we were revising genera. For those of you who don't know what a revision is a kind of work where you bring together everything that's known about a taxon. You produce a new classification, you provide identification tools, describe species, provide phylogenies, distributions, basically everything. Revisions are the cornerstone of biodiversity work. If a taxon's never been revised, you can't do anything with it. You can't name them. You know nothing. So revisions are really, really important. And this huge number of specimens available to us has allowed fairly comprehensive revisions. And the one I'm referring to here, a very recently completed revision of Scleroculus, was based on 13,000 specimens. And it allows us to say that based on this good coverage, we're pretty confident this genus, which ranges from Canada to Argentina and on to a few oceanic islands, has 58 species, and 54 of them are new. And this is an important point because it's a hard number that says, yes, we've actually counted the species, and yes, the novelty rate, the proportion of undescribed species, is really as high as everybody keeps predicting. So we've had this kind of number over and over for dozens of revisions. And here's just a couple more examples. solid evidence that indeed our estimates of species diversity are more or less correct at 80 to 90% more than known species. So that's it for the Sphaeroceridae. That's it for the top 20. Before I wrap up, let me leave the top 20 and mention that in my lab, we've also looked at a number of other families outside the top 20, and we're finding almost the same novelty rate in those other families as we did in the top 20. So the Clusiidae is an example, and those funky things in the front of the head are cheek jowls that the males use to battle for territory. But I'm not going to talk about Clusiids. I'm going to talk about just one more family, my favorite family at the moment, the family Micropicidae. The Micropicidae are known as stilt-legged flies for obvious reasons. This is an exceptionally long-legged species from Madagascar. But Micropicidae, in contrast to the dull Sphaeroceridae, are big, colorful flies. They've attracted a lot of attention. And one would think that the novelty rate would drop in this group because they're kind of the butterflies of the acalyptrate world. But in fact, the great majority of Micropicidae are undescribed, and many of the genera aren't described. Some of them are colorful mimics of wasps. Some of them have really interesting courtship behavior, mating behavior. Some of them have interesting host-seeking behavior, sticking their heads down burrows, oviposition behavior. They're interesting flies. So you'd think they'd be well known. The first genus of Micropicidae that I decided to revise when I was first starting to work on the group was the genus Mesoconius. And these are big, gorgeous flies, and they have attracted some attention in the past because they're restricted to high alpine localities, and they tend to have narrow ranges. They're important in conservation biology. And the genus is well defined. It's got good characters, easy to recognize. So I thought, this is an easy revision. But this turned out to be a new species. This turned out to be a new species. This turned out to be a new species. And these new species aren't just based on these nice colors you're looking at. They're based on dissections, sequences, all evidence. This turned out to be a new species. This turned out to be a new species. Then you're getting the picture of this one too, and these two. Even though this should be one of the best known families of acalyptrate diptera, 37 of the 58 species were new. I divided my revisionary work on this group into two parts, Central America and South America. This beautiful fly, Mesoconius tigrinus, one of my favorite Mesoconius, was a new species from Costa Rica. And this kind of work, taxonomic revision, should allow anybody else to come along once a revision is done and identify their species, because there's always identification tools in a revision. And furthermore, it should allow any further new species to be easily recognized in context against that revision. And I had a chance to sort of test out that claim myself just this last year when I was in the Ecuadorian Andes. I collected a whole pile of stuff, including several Micropicidae that had been described in that revision several years earlier. But I also came across this really stunning fly, a very large fly, which I immediately recognized as a Mesoconius that wasn't in my revision. So I was able to say, this is actually quite unusual. In the field, aha, I found the new species. Now, you'd think that would be a commonplace thing, given that most insect species are undescribed. But in fact, it is quite rare. Most insect species are described years or decades after they're collected and after years of work in the lab. So for me, despite the fact that I have discovered and described lots and lots of species, for me to encounter this thing in Ecuador and go, yes, I recognize it, it's a brand new species, was really pretty exciting. I hope to discover it, I hope to finish it I hope to describe it formally in a publication soon, but, well, so many species, so little time. That applies at so many levels. I'm out of time for this lecture. There's too many species, too little time for the taxonomic community to deal with, which actually we can put that. And it just applies at all levels. And I think that's probably a good place for me to stop. So I think there's probably time for questions.
What distinguishes a fly from a beetle from a wasp?
Okay, what distinguishes a fly from a beetle from a wasp? That's a nice, simple question. As I said, flies, as the name Diptera indicates, are the two-winged insects. So on the easiest level, count the wings. If it's two, it's flies. If it's four, it's a wasp or a beetle. Of course, there's lots of other differences. Flies usually have non-biting mouth parts, so you don't see mandibles, whereas in beetles and wasps, you'll see mandibles. Flies are usually softer than the others, so I always say, look at the goo on your shoe, and you can tell by how hard it is which order it belongs to. Maybe not a good recommendation. So flies are easy. Beetles are easy too. If you see a straight line down the back, it's a beetle, because the forewings of beetles, called elytra, meet on a straight line down the back to form a shield or cover over the body. So that straight line is a dead giveaway. So there's the flies, there's the beetles, what's left is wasps. Okay, is that fair enough?
And what's a midge?
A midge, that's an interesting question. The term midge refers to a number of small, delicate kinds of flies. They're what we dipterists refer to as the lower Diptera. They used to be called the suborder Nematosera. So it's a loose term referring to small, delicate Diptera. Yes?
Question about sort of being able to identify species. Because what you've been indicating in this is how so many of these new species are, I guess, undiscovered or unclassified. Would you say that one of the major ways of identifying these species is genetic sequencing? Is that my understanding?
Okay, my hearing isn't that good. I may have missed some of what your question was, but I gather you were asking me, what are the main techniques used to recognize a new species?
That's right.
Yeah. Okay, well, there's a lot of answers to that question. It depends upon who you are, what lab you work in, and what taxon you work on. And it depends on whether you mean recognize, diagnose, or define. I mean, these are not all entirely the same things. I would say the great majority of insect species are now first recognized and described and characterized first and foremost using morphology, primarily male genitalia, but in some groups like the micropesids, the internal female genitalia is incredibly complex and full of good synapomorphies characters. Molecular characters are valuable. So I work somewhat differently than many people. I'll work with morphology first, and then I'll come in and see if the molecular data supports or conflicts what I've done with morphology. In most cases, it's supportive. Every now and then, the molecular data has allowed me to go, oh my goodness, there was two species here, I better look again. And sometimes it's allowed me to go in and match males and females that I couldn't have matched before. Like sometimes males and females get described as different species in the past, and it's hard to link them up. So yeah, it's of value. And then of course there's the question of cryptic species, which is a bag of worms that I wanted to keep tightly sealed off today because it's a very wiggly one, but it is an interesting one. Yeah.
How many species have you named, and have you named any of them after you?
Oh no, that would be in the worst of taste. It's been done. There's actually Percy Moore, many years ago, named a leech Percy Moorensis, but that was considered to be an example of bad taste in insect taxonomy. No, patronyms are common. We often name taxa after the collectors or people who have done work in the group, but I don't know anybody who's actually named a species after themselves. That'd be so tacky. Yes.
Collecting. You do a lot of collecting in foreign countries. Has that become more difficult over time? Are countries becoming more proprietary over species that you find in their country?
Absolutely, yeah. That has become a huge problem. The question, if you didn't hear it at the back, was: has international collecting become harder and harder over the years? So it's essentially the question. And absolutely. I'm very fortunate because the the massive coverage that I have of the New World, Central and South America, developed from pan traps, was done for the most part before the current regulations came into place. I still have a standard practice of sending my type specimens back to the country of origin to maintain good relationships. But those collections were largely made before the current regulations came into place. Now, when I go collect in a foreign country, my paperwork is just horrendous. But that said, it's doable. I work closely. I prefer to work in Ecuador right now because I'm a research associate of the Biodiversity Institute there, which smooths my path. And I've had associateships with Costa Rica and various African countries. It's doable. It's just a lot of paperwork. There are a few countries where it's impossible. I'd love to work in Indonesia, but it just isn't happening. I know that some people here at ROM have been successful in working out arrangements and have gotten permits. I've never been able to do that. And there's a couple of other countries where I haven't been able to make arrangements. But for sure, it's a lot more complex than it ever used to be. Yes?
So in New York, basically the spotted lanternflies took over in the past 5 years.
Yes.
And biodiversity is constantly changing with the climate. I guess my question is, how quickly will it take for them to get here potentially and become a Canadian club? Like, how do we change our national numbers of biodiversity to account for these?
No, that's an excellent question. How do we keep tabs on how our biodiversity is changing with new introductions and ranges that are shifting north with global warming? And it's a really important question. We assume that the Canadian fauna is about 5% exotic. That's just a rough guess, because it's sometimes very difficult to say when you look at a whole Arctic species, a species that's in Europe and in Canada, and say this is an introduction, because you don't know. Maybe it was here all along. If it's easy if something has just appeared within our memories, sort of the seven-spotted lady beetle or the European paper wasp that I mentioned earlier in my talk. We know exactly when they appeared here. But sometimes we don't know. And as for something like the spotted lanternfly, well, that's a nice easy one, because the spotted lanternfly is so huge that the iNaturalist community, I don't think is going to let it move a mile without more records. Beautiful insects and terrible prolificous pests. I don't know whether I know it's been recorded in Ontario, but I don't know whether it's really going to take a foothold here. I don't have any real insight into that. So I'm not sure if that answers your question or not. Okay, right here.
Yeah, I was going to ask, how helpful do you think that community science on platforms like iNaturalist is for documenting, for example, Terra, when we talk about the diversity of species, recognizing that you need samples quite often to do identification. How helpful are those programs become?
Yeah, that is a really interesting question. How useful are platforms like Bug Guide and iNaturalist in documenting diversity? I think they're fantastically useful, but of course they are limited to the 20% or so of taxa that are potentially identifiable from photographs. Maybe it's a lot more than 20%. I've actually been really surprised by the success that some Bug Guide contributors have had in photographing some of these tiny, really difficult groups of flies and getting those photographed, identified, getting them up online. So maybe the number's huge. I'm not sure if you know the journal, the Canadian Journal of Arthropod Identification, but it's an open access journal which was deliberately developed, specifically developed to address that issue, to provide open access tools, photograph-based tools that people could use to actually key everything. and it's moving in that direction. So yeah, I mean, these platforms I love them. I use them all the time for larger things. It's faster for me to look up something on Bug Guide or iNat than it is for me to go to my texts. And they're pretty good. I'm impressed.
Quick
Where are we? Over here.
follow
All right, I got to tell the story of Thydia moronii. I discovered a spheroceriid in small algal strands along the shore of Meech Lake. Meech Lake Accord, okay. So it was a new species, nice new species, and I was revising this genus Thydia. And I went, well, gee, I'd like to draw our Prime Minister's attention to the biodiversity crisis and how we really need support for taxonomists. So I kindly and generously gave a patronym to Moroni. I called it Thydia moronii. And I sent a letter in to him about biodiversity. It was all meant in the best of spirit. And I got a letter back from the Prime Minister's office that basically said, oh, go away, don't bother us. And I forgot all about it. And then sometime later, a newspaper article appeared. Canadian entomologist named scum-sucking fly after Prime Minister. Thanks, Rob Ruffley. Yeah. Okay, over here.
I think at one point you stated that there was a certain species that was totally eradicated?
Yes, well, extirpated, not eradicated. Eradication is difficult. Extirpation is unfortunate. Yeah, that was the Necrophorus americanus. It's the American burying beetle, and it just disappeared about the 70s. There's a lot of speculation about why that happened. One of the common theories is that it was in rapid decline once the passenger pigeons were gone, because it's associated with carrion about passenger pigeon size. Another theory is that the large numbers of predators like coyotes outcompeted them for small carrion, but whatever, they're gone. Yeah, you're right. It's not at all unusual for ecologists and taxonomists to say, "Wow, we haven't found this in 100 years. It's gone, bummer." And then 2 years later, someone finds one, which is great. But in general, when we declare something like Necrophorus americanus as extirpated, we've looked at all available collections. We've gone out and put carrion traps out. We've searched. And we've concluded on the basis of pretty good evidence that, yeah, it's gone.
After 5 years, 10 years?
I can't tell you that. Yeah. I've got a question right there. But someone might have to shout really loud because my hearing's not very good. Here first. Do I use AI to help catch the bugs? Is that what you're asking? To identify them. Well, that's a really good suggestion, and I understand that a lot of people are working on that, but I'm not. No. Over here, yeah.
Hi, Steve. Great talk.
Thanks.
About micropeaches. So I noticed that a lot of them are holding their forelegs over their head. Is that to mimic long antennae?
Yeah, that's the usual theory is that they're trying to be as much like wasps as they can. So they do wave them up and down like that. Yeah.
Is there a specific group of wasps that it's mimicking, or just?
Well, they usually mimic Ichneumonidae, which may surprise you because, of course, Ichneumonids aren't normally thought of as stinging Hymenoptera. But the models in this case are Ichneumonids that can poke you with their ovipositor. So they are stinging Ichneumonids. Yeah. Well, that's a really good question. The question is, do flies only mimic Hymenoptera? And of course, the great majority of fly mimics are mimicking Hymenoptera because Hymenoptera stink, And so they're trying to be fakes. They're trying to pretend that they're well defended when they're not. But you're absolutely right. There are lots of wasps that mimic other things. Lots of flies that mimic other things for various reasons. There are some flies that are dead ringers for beetles. They look exactly like beetles, presumably because those beetles aren't tasty and they want to look like something that's not tasty. There's some flies that I've studied that looks so remarkably like beetles. I can't tell the difference until I actually catch one and put it in a net. So yeah, there are some interesting exceptions, but usually they pretend to be stinging insects. And I got a question right up the front here that I'm sorry, yeah.
So speaking of species, mutation stacking doesn't do just what I'm trying to explain, but to some extent, and the reason for diversity between species can be, if dumbed down enough, be referred to as just mutation, or for the matter, genetic variations, their default laws, stacking up. At what point can we call a species unique?
Okay, that's a really good question that I'll try and summarize as what are the criteria we use to decide that a species is new? What's the cutoff to decide whether a species is new? And is that fair enough?
Yeah.
Okay.
When can we say it's not enough to just call this a species in this genus? It's not enough to call it just a sentence. It's all.
Okay. I'm not sure I got that entirely, but let me try and answer it anyways by saying that species are real, for starter. So even if we aren't able to recognize what makes a species, they know, right? So that might be one of the reasons that we often turn to something involved with their own mate selection, like male genitalia. And so male genitalia are often used to decide whether there's one species or two. If the male genitalia are different, they're different species. Not always, but that's typically very useful. Is that sort of answering the question?
Yes, that does answer my question.
Oh, okay.
Speaking of reproduction, what stops inbreeding from being, if you refer to the growth of a species as advantageous, then what stops inbreeding from being advantageous in itself?
Yeah, I don't think I can answer that question. Okay. Thank you very much.
Thank you, Steve. That was a wonderful talk, and I can see why so many of your students continue studying Diptera after they leave your lab. Your passion is contagious. I think there's a lot of people here that now may look at flies a little bit differently than they have previously. I certainly will, and I'm very happy to be contributing to your micropizid edition with the loan I prepared yesterday for you. So hopefully there'll be some new species there too. So thank you. I would like to thank Robert and Jane Hess again for their support and sponsorship of this talk. Thank you. I would like to thank ROM Programs for their assistance. Aaron Kerr at the back has been very, very helpful, along with our video crew who's recording, and the audio team. For those of you that are TEA members, great. For those of you that are interested, the membership is $30 a year for an individual, $35 for family, and it's free for students. So if you're a grad student or undergrad student or even high school or elementary student, it's free to join the TEA. And if you don't want to join, but you're interested in learning about the talks that we have a friends list. So you can receive messages about when our meetings are. And it's actually, they're open to anyone of the public. So if you're interested, please join our list. Our membership person is not here today, but it's pretty easy to find out how to join. The ROM membership, since we're at the ROM, is $125 a year. The family is or a dual membership is $225. And there's also different rates for students and for seniors. So if that interests you, please join the ROM. Because this is a TEA meeting, I have a few announcements about our next meeting and some other activities that are happening. At the ROM, we have an ROM Ideas Symposium, which is open to the public, and that's going to be on March 2nd. So it's a ways away, but it talks about some of the research that's happening here. Wildlife Photographer of the Year, which I'm sure many of you have seen before, is December 21st opening. TEA members have a promo code for the ROM. I encourage everyone to go see those because it's a fabulous exhibit. Nature in Brilliant Color is opening December 14th, and it's a very visually stunning exhibit. And our next TA meetings are January 25th, and it will be Michelle DeLeo. She's a research scientist with the Ministry of Natural Resources and at Trent, and she's going to be talking about butterfly surveillance. On February 22nd, the ROM's Brad Hubley and Michelle Locke from the Canadian National Collection will be talking about capturing museum specimen data using public participation platforms. So that's going to be an interesting talk as well. On the 22nd of March, we have the Student Symposium, which is always great for young graduate students or even undergraduate students to present some of the work that they're doing. And in April, we have a Bug Rearing Day, which is often takes place at the zoo, at the Toronto Zoo. They host it, which is great. We don't know what the date is yet for that one. It varies. So I'd like to thank all of you for coming this afternoon. I think it's been a wonderful way to spend the day. And for the members of the Toronto Entomologists Association, there will be a reception up in the glass room immediately following when I stop talking. Okay, thanks everyone. Nope.